Today's lecture covers molecular biology. This area of science concerns biological activity at the molecular level. This video is located and can be viewed under optional resources located below. Protein synthesis means to make a protein. On the next several slides, we'll look at how that process occurs. We will begin protein synthesis with a quick look at DNA. DNA stands for deoxyribonucleic acid. The building block of DNA is a nucleotide. The job of DNA is to direct the replication or the copying of proteins. A gene is a portion of DNA that carries instructions for making one protein. The order of the bases on the DNA ladder determines which amino acid is put in the protein chain at each point. You can see the DNA ladder in the picture on the right. I mentioned that the building block of DNA is a nucleotide. A nucleotide has three parts: a phosphate molecule, the sugar deoxyribose, and a base. DNA is named for the sugar it contains, deoxyribose, and that's where the name DNA or deoxyribonucleic acid comes from. Let's look next at the bases. There are 4 DNA bases. They are adenine, represented by capital A, thymine, represented by capital T, guanine, represented by capital G, and cytosine, represented by capital C. Although these bases occur in very long chains, they are read in triplets known as codons. One codon matches a particular amino acid, and we'll take a look at that in just a minute. This drawing represents a DNA ladder. The sides of the ladder are made up of phosphate and sugar, and remember it's DNA, so the sugar is deoxyribose, and the rungs of the ladder are made up of the bases A, T, G, and C. Take a look at this part of the DNA ladder that has been flattened out so you can see it a little better. There is always a pairing of the same 2 bases. A and T always pair, and G and C always pair. And always there are 2 hydrogen bonds between A and T, and 3 hydrogen bonds between G and C. Here is a portion of the DNA ladder showing one of the bases on each rung. Can you predict what base should pair with each of these? The answer is on the following slide. Remembering that A and T always pair and G and C always pair, the sequence should be AT in the first rung, CG in the second rung, GC in the third rung, and TA in the fourth rung. Here is a recap on some of the important points I've mentioned. In the DNA ladder, A and T always pair and G and C always pair. Hydrogen bonds connect the 2 bases to one another. There are always 2 bonds between A and T and always 3 bonds between C and G. The sides of the DNA ladder are made up of alternating sugar and phosphate groups, and remember the sugar is deoxyribose, and the rungs of the ladder are made up of the 4 bases and are part of the ladder that change. The sides do not change, but the bases of the rungs do change. DNA contains the message of how we make a protein. The problem is the proteins are made at the ribosomes, which are out in the cytoplasm, and we need to get the message from DNA out to the ribosome. DNA is too large to leave the nucleus. The holes in the nucleus are very small. So what we need to do is make a copy of the part of the DNA molecule that tells us how to make a particular protein. This copy is called mRNA or messenger RNA because it carries a message out to the ribosome of how to make a protein. Messenger RNA is much smaller than DNA and is able to leave the nucleus through the pores and go to the ribosome with the message. Like DNA, RNA also has 4 bases. One base is different, however. RNA has the bases A, U, G, and C, adenine, uracil, guanine, and cytosine. The one that's different from that of DNA is uracil. In RNA, you'll find the base uracil in place of thymine. Thymine is found only in DNA and uracil is found only in RNA. The other 3 bases are found in both. When we make a protein out of amino acids, we're going to use 3 different forms of RNA. One is messenger RNA, and we've already talked about that. The job of messenger RNA is to carry the message from DNA, which is in the nucleus, out to the ribosome. rRNA is ribosomal RNA, and ribosomal RNA forms part of the ribosome. tRNA is transfer RNA. The job of transfer RNA is to carry amino acids that are in the cytoplasm to the ribosome, where we build a protein out of them. Remember that a protein is a long chain of amino acids, and when we make a protein, also known as protein synthesis, there are two important processes that are involved. One is transcription, which is the actual copying of messenger RNA from DNA, and the other is translation, which is interpreting the message on messenger RNA at the ribosome and making a protein out of amino acids. The first process in protein synthesis is transcription. It is the process in which a part of the DNA is copied onto a strand of messenger RNA. Messenger RNA then carries the message of how to make one particular protein. Transcription occurs in the nucleus because DNA is too large to get through the pores and leave the nucleus. That's why we need to make a copy of the part of the DNA strand that tells us how to make that protein. Here is a picture of the process of transcription. You can see that the two strands of DNA have split, and one of the strands will be used as a template to make messenger RNA. So we're going to copy a portion of one side of the DNA molecule and produce messenger RNA. Here is the DNA ladder. Remember, it is composed of two strands. The two sides will split apart, and a portion of one of the sides will be used as a template to make a messenger RNA strand and take the message of how to make that protein out to the ribosome. The first thing that has to happen is we have to break the hydrogen bonds between the bases. Remember, A and T are always paired, and G and C are always paired. Here again are the two sides of the DNA ladder. Remember that when it splits, one of the sides will be used as a template to make a strand of messenger RNA. In this case, we'll use the bottom as the template. On the bottom is the DNA template. On the top is the strand of messenger RNA that we're making to take a message out to the ribosome of how to make this protein. Remember that RNA contains uracil instead of thymine. If you will click on the link Animation of Transcription, you'll be able to see what transcription looks like. Also, on the following slide is a video of transcription. This video is located and can be viewed under optional resources located below. The second process is translation. We have to interpret the message that's on messenger RNA so we know how to build the protein. Here is the process of translation. Messenger RNA inserts itself between the two spheres of the ribosome. The message is then read by the ribosome in triplets. So the first triplet is A, C, G. A triplet is known as a codon. The first codon again in messenger RNA is A, C, G. Transfer RNA, which carries amino acids, has an anticodon that must match up with the codon on messenger RNA. The anticodon on transfer RNA is UGC. A and U pair, G and C pair, C and G pair. That's the first codon and anticodon. Attached to the anticodon was the first amino acid on transfer RNA. The second codon is red. The second codon is UAC. The anticodon that would pair with that is AUG. It's at the top of the second transfer RNA carrying an amino acid. Once the second amino acid is in place, a peptide bond forms between the two, and this is how the amino acids will be added and a long protein formed. A codon has 3 bases along messenger RNA. An anticodon has 3 bases along transfer RNA that pair with the codon. The peptide bond is the bond between 2 amino acids. Click on the link below, Translation Animation, to view the process of translation. Here is a link to the process of protein synthesis. This video is located and can be viewed under optional resources located below. Here are a few more important terms to know. DNA replication is the copying process of DNA before cell division. A mutation is when a change occurs in the sequence of DNA bases, and it is usually lethal. And a mutagen is either a physical or a chemical agent that causes a mutation. Mutations induced by chemicals often lead to cancer. The information on all these slides is important, but as you review, be sure that you go over these points. Know the definition of transcription and where it occurs, the definition of translation and where it occurs, the functions of the 3 types of RNA, what is a peptide bond, why is messenger RNA produced, and what parts of the DNA ladder can change.